After the horse, one of the most important animals to be domesticated is the camel, especially in North Africa. But people don't understand that the camel actually originated in the Americas and migrated through Asia all the way to Africa and the Middle East. But when did it first get domesticated? Well, because the camel is associated with the Middle East because of its wide use, understandably, it's, a desert. So why wouldn't you use an animal that stores water and can survive many days without it? The scorching heat of the desert does not play with anybody. And a lot of people have died, a lot of animals have died, and through the process of elimination, a lot of animals have been removed from practicalness in the desert. So the camel, though suited for the desert, might not actually be a desert animal. Well, at least not according to some studies. So if you look in the Middle East, you see a lot of diversity in, the camels. It seems like there's a lot of interbreeding and inbreeding and stuff like that that leaves a genetic footprint. But if you go to Somalia, the country of Somalia, you start to recognize something that's strange, which is a uniform of genetics when it comes to the camel, which makes it seem like it's not as, you know, diversified as outside of Somalia, which gives some people, some geneticists, the indication that Somalia might be the foundation of domestication when it comes to the camel or dromeda. As we read, the dromedary is one of the largest domesticated domesticates substantially sustainably used in arid and hostile environments. It provides food and transport to millions of people in marginal agricultural areas. We show how important long distance and back and forth movements in ancient caravan routes shaped the species. Genetic diversity using a global sample set and ancient mitochondrial DNA analysis, we describe the population structure in modern dromedaries, dromedaries, and their wild extinct ancestors. Phylogenetics analysis of ancient and modern dromedaries suggests a history of restocking from wild animals from the southeast coast of the Arabian Peninsula. Dromedaries now extend the list of species for which classic models of domestication from a single center and from wild conspecific individuals in isolation are rejected. The final part says this: dromedaries have been fundamental to the development of human societies in arid landscapes and for long distance trade across hostile hot terrains for 3,000 years. Today, they continue to be an important livestock resource in marginal agroecological zones. However, the history of dromedary domestication and influence of ancient trading networks on their genetic structure have remained elusive. We combined ancient DNA sequence of wild and early domestication dromedary samples from arid regions with nuclear, microsatellite, and mitochondrial genotypes information from 1,083 extant animals collected across the species range. We observed little geographic signal in the modern population, indicative of extensive gene flow and virtually affecting all regions except East Africa, where dromedary populations have remained relatively isolated in agreement with archaeological findings. We identify wild dromedaries from the southeast Arabian Peninsula among the founders of the domestic dromedary gene pool. Approximate Bayesian computations further support the restocking from the wild hypothesis, with an initial domestication followed by introgression from individuals from wild, now extinct populations compared with other livestock, which show a long history of gene flow with their wild ancestors. We find a high initial diversity relative to the native distribution of the wild ancestor on the Arabian Peninsula, and to the brief coexistence of early domestication and wild individuals. The study also demonstrates the potential to receive ancient DNA sequences from SES remains excavated in hot and dry desert environments. And here are the conclusions. Examining modern genetic diversity and its global distribution, it is possible to gain insight into the domestication process because the absence of recurrent introgression populations close to putative domestication centers are assumed to retain higher levels of ancestral polymorphism. Such distribution of genetic diversity has been suggested to explain the frequency observed negative correlation between genetic diversity and geographic distance from the place of origin in numerous livestock species. In the case of dromedary, before the introduction of domestic form, there had been no representatives of camelis on the African continent since the late Pleistocene, and the Holocene. Native distributions of wild dromedaries seems to have been restricted to the Arabian Peninsula. Modern dromedary populations from the Arabian Peninsula therefore were expected to display the highest levels of genetic diversity and variation. To test this expectation, we combined two comprehensive data sets encompassing 759 mitochondrial data sets: Eastern African, Western and Northern African, Northern Arabian Peninsula, Southern Arabian Peninsula, and Southern Asian, including Australia. The shared genetic diversity and population structure in modern dromedaries, in contrast to the hypothesis that the greatest ancestral variation is retained close to the area of domestication, we observe similar amounts of zygosity and allelic richness among the different populations. This finding precluded any conclusion about the existence of an ancestral population or a geographical center of dispersion. For comparison with other camelids, shared diversity also was revealed by the analysis of molecular variants with 95.7% nuclear and 95.3 mitochondrial DNA of the variation distributed within populations. Hence, we investigated genetic population structure in modern dromedaries, disregarding their geographic origins. Mitochondrial median joining network analysis split the 76 haplotypes into two haplogroups, HA and HB, containing 6 major haplotypes. This partition was supported by Bayesian phylogenetics analysis. No phylogeographic pattern was detectable because the 6 major haplotypes were observed across the global range of the species. In contrast with the nuclear structure analysis, we retrieved an optional number of 2 ancestral populations, clearly separating EAF dromedaries from all other populations. This separation also is reflected in the 3D factorial correspondence analysis and limited population differentiation, a plausible consequence of the intense back and forth movement that characterizes the use of dromedaries in cross-continental trading. EAF, by the way, is East Africa. So their conclusion is that the camels must have come from East Africa, picked up at some point by the Middle Eastern population, and then moved back and forth between the continents until they became ubiquitous with the Middle East. And of course, they weren't hugely adopted in areas where they don't bode that well, like Europe. But it is a surprise. Genetic distinctiveness of East African dromedaries. Modern East African dromedaries exhibit the lowest nuclear but the highest mtDNA diversity of all populations. These elevated values could, in principle, be explained by a large proportion of ancestral diversity in the mitochondrial DNA or by a cryptic population structure not accounted for in the analysis. Although 85% of the investigated haplotypes belong to HB, dromedaries in Eastern Africa exhibited a more balanced ratio of HA 38% and HB 62%. These results may be interpreted as the consequence of a random founder effect followed by a successive gene flow with a restricted number of sires. Globalization of genetic diversity might not have affected the East African as much as the other populations, likely because of its isolation from the northern part of the continent by eco-geographical obstacles. Ethiopia plateau and the swamps of the Sud physiologically constrains humidity, food plants, lack of salt, disease, and perhaps most importantly, cultural barriers. To investigate subtle population structure that might have been masked by the high distinctiveness of East Africa, we excluded the later from structure analysis and observed 9 independent clusters. Despite substantial admixture to dromedary population from an isolated mountain region in southwestern Saudi Arabia, segregated dromedaries from Oman and UAE separated from the cluster containing Southern Asian individuals, whereas WNAF and NAP populations shared common ancestors and genetic diversity. Within the latter, only the Hadana breed appeared to have contrasting genetic makeup. Introduction of Arabian dromedaries into Africa. The absence of genetic structure between WNAF and NAP points to an extensive exchange of dromedaries introduced into northern eastern Africa from the Arabian Peninsula via the Sinai, possibly starting in the early millennium BCE and intensifying in Ptolemaic period. From here, dromedaries spread across northern Africa, but their adoption into local economies may have been slow, considering that the first unequivocal evidence for the presence in northwestern Africa comes from archaeological layers dating to the 4th and the 7th century CE. Although WNAF NAP showed close cross-continental affiliations with Southern Arabian and Asian dromedaries, the two African populations were genetically the most distinct of the ones that are tested. Now, I don't want to mislead you into thinking that this is conclusive, but it also does show that the domesticated ones in East Africa is the most unique of the others, and the others seem to have been wild and then they were brought to East Africa, possibly due to, hey, you guys domesticate these animals, we have these animals, trade, they interbreed with some of the East African ones, and because of their usage, they're exported during the Ptolemaic period of ancient Egypt and then into North Africa. And just like a lot of animals, chimps and stuff like that of East Africa, humans, the complicated history clouds the judgment of the geneticists and everybody really. But it seems like East Africa might be the hub of domestication of camels. Not where camels came from. Keep that in mind. Separate that from mind. Camels come from America. That's why you have llamas in America that are related to the camel.